Hello, everybody, and welcome to the start of full distance learning. We're going to start off today for many of you with an at-home lab. A few of you didn't get an auger plate from me, and so you are just going to watch a slightly longer version of this video and use my experiment to collect your data. But those of you at home, I'm just going to explain how to set this up, and then you can grab your supplies and you can go and do this yourself. We are now officially moving into Unit 4, which is about cell types and cell structures. We started this a little with cell membranes already, but now we are going to continue and move into this in a little more depth. So cells, as you know, are the basic structural and functional units of life. They're the smallest thing that are able to carry out all the characteristics of life on their own, and all living things are made up of cells. So before we get started today, I want you to take a minute and think about what you know about cells. Grab your notebook and draw two cells, one that is prokaryotic and one that's eukaryotic. Don't look anything up. This is just kind of a little pre-test for yourself to see what you remember. We're going to come back to this more tomorrow and talk about these two cell types. As you draw this, notice what parts or what characteristics these cells share and how they're different. The lab we're going to do today looks at how the size and shape of a cell affects the rate of diffusion. You probably know that most cells are very small. They're microscopic, and some of them are exceptionally small. And the reason that cells need to stay very small has to do with how quickly materials can diffuse in and out of them. And the shape of a cell also affects how quickly that process happens. And that's what we're going to be looking at in this lab today. Remember, we talked briefly last week about the fact that diffusion is a type of passive transport where molecules move down their concentration gradient. In the lab today, you are going to be putting some blocks of agar, that'll be a model for cells, into some vinegar. And the vinegar is going to be diffusing into those cells. So that's what we're using to kind of model molecules diffusing into a cell. So as you do this, remember to think about what this is representing. Cells obviously don't need vinegar, but what other things do cells need that come into the cells by diffusion, and what leaves cells through diffusion? And why is this process so important? So I'm going to switch over here to actually a Google Meet just so that you can see me. Hi, everybody. Hope you're having a good day at home. But I'm actually going to move my camera down here so that you can see the setup for this lab. So here's my little plate of agar. I've actually already cut a little piece out of here just because I had to test something and make sure this was working. You can go ahead and cut that open. What I would recommend to get this out, I have a cutting board below me here, so make sure you're cutting on a surface that will work for you. It will work easiest if you just kind of run your knife along the side of the agar to kind of loosen it up. And then you can even use the knife on the edge. You should be able to kind of slide that whole block of agar right out of the dish in one big chunk. From there, you can cut it into cubes and rectangles of various sizes and shapes. There are some guidelines in the procedure of the lab to kind of help you with this. One thing to remember, you may be as creative as you would like with the shapes that you make out of your agar, but you do need to be able to calculate their surface area and their volume pretty precisely. So it works best for this lab if you use things that are cubes or rectangles. It's also helpful if you have a couple that are roughly the same shape. So I'm trying to make these mostly a cube. That one looks pretty close to a cube, I think. A little bit smaller. But have them of different sizes. So I'm going to have kind of my larger cube here, and I'm also going to cut a smaller sized cube. That will allow me to see how the size alone affects the rate of diffusion, even if they're the same shape, even if they're both cubes. So there, I have a big cube and a little cube, and this is just all my extra over on the side. And then I'm also going to cut out a couple of other shapes. I'm going to cut myself kind of a big, wide, rectangular solid, something like that. And like I said, you do not need to cut exactly the same shapes as me. You are welcome to be as creative with this as you like. I'm going to cut one that's maybe flat and rectangular, but a little bit thinner, something like that. And maybe I'll cut one that's really long and skinny. You need to have a minimum of 3 blocks of auger that you're testing, but you are welcome to cut as many as you would like out of the auger that you have available to you, because this is kind of cool and fun to watch. All right, so that's all my extra. I'm going to cut this one off just so the edges are a little more square. As you calculate surface and volume, or surface area and volume, even if your shapes are not perfectly cubical or perfectly rectangular, you may measure them as though they are. It's okay if there's a little bit of error in here, you will still get the point. So this is all extra over here. So now you need to take some very careful measurements of all of your different shapes, because like I said, we are going to be measuring surface area and volume. So I'm going to take just a minute here and take some measurements of my shapes. And then I'm just going to put them into this container. You can use any container that you have available. It is most helpful. You do need something where the bottom is either clear, so you can put it on a white surface, or has a white interior. You will not be able to see this if you use like a coffee mug that's dark brown on the inside. So you need something either clear or with white on the inside. So if you have a bowl that's white, that will work just fine. I'm using clear glass so that you can see it on the video a little better. So I'm going to make some measurements. I'll come back in just a second when I have them sitting in here and we're ready to start timing. Okay. All right, so I have measured the length, width, and height of all of my shapes and recorded all of that data. Then I placed all of my little cell models into this container. The next thing I'm going to do is add some vinegar. If you don't have vinegar at home, you can try this with Sprite, or a flavored bubbly water would work best because they're clear. Possibly lemon juice or lime juice would work, but you need something that's acidic, but that you can see through well enough to see the color changes that are going to happen. For those of you who are collecting data, make sure you have a stopwatch ready because you are going to be starting your timer when I add the vinegar. You want to have all of them in there at once and start your timer when you pour the vinegar on. And what you're watching for is you're keeping track of the time it takes for the entire shape to turn yellow all the way through. So I'm going to start this one and you can kind of watch to see what I mean. So if you're starting your timer, you're going to start it in 3, 2, 1, go. You need enough vinegar to cover all of your shapes. And then you want to just move them around so that they're not touching each other. Mine aren't, but I also want them moved out where you can see them all. And you will soon be able to see that the edges of them, hopefully you'll be able to see this in the video, the edges of them are starting to turn yellow already. And as the vinegar diffuses into them, the cubes will turn more and more yellow. To you on the video, they might just look like they're kind of disappearing, but you are going to time how long it takes until there is not even a speck of the dark bluish green color left in the middle, and they are completely yellow all the way through. The lab document for this activity is posted under the homework for this week. It has the procedure for you and a data table to record your results and the discussion questions to think about afterwards. So go cut out some fun creative cell shapes, find some vinegar, and have some fun doing some science at home. Have a great day. For those of you going to do your own lab, if you're collecting data from the video, stick around a little bit longer and watch till these guys turn yellow. Have a great rest of your day, folks. Those of you who are sticking with me for a while, you can start to see the edges of these are getting all yellow. They're really pretty. I will tell you when each of them completely changes color. So we have here a very tiny cube. That's my small cube right there. This is the larger cube that I made. So those two are the same shape, but different sizes. We have kind of a smaller sized rectangle here and a larger rectangle. I don't know if those are exactly proportionate, but they're similar. And then one very long, skinny rectangle, just so we can look at a few different shapes. And I will let you know when each of these turns completely yellow through the middle in case you can't catch it on the video. Make sure to record the time for each of those so that you can save that data in your data table. The long, skinny one's getting pretty close. It still does have a hint of blue there in the middle, but we'll keep an eye on that one. Just about there. Still the faintest hint of a dark blue line that I can see right through the middle. I know that's hard to see on the video. All right, long, thin rectangle is done now. I think you can see on the video this little guy's getting close. There's still a teeny dot of blue in the middle. Keep an eye on this little one. There's still just a dot of blue in there, but it won't be too much longer. We're just waiting for the last little pinprick of blue in this small cube. All right, let's see here. Last little bit in the small cube is fading away, and it is all the way yellow. Let's see, right about, come on. Takes a long time for that last little bit to go. right about now. You can count this small cube as all yellow. Looks like the next one might be our smaller rectangle. There's still a strip of blue in the middle that's fading away, but that'll probably just be another minute or so. So this smaller rectangle here is just about done. There's a hint of blue still in the middle, so I will tell you when that one is finished, but it's very close. All right, this smaller rectangle you can count as done right about now. We are just waiting for 2 more of our cells to finish turning all the way yellow, waiting for that vinegar to diffuse all the way to the middle. Our larger cube and the larger rectangle are the 2 that are left. So our larger cube is getting pretty close. There's still a dot of dark blue in the middle that you can kind of see there. All right, our larger cube here is getting close to finished. It's not quite there, but it's close. The largest cube still has just a faint dot of dark blue in the middle, so I will tell you when that one's done. Just one last little dot on this big cube. We're almost there, but keep an eye on those timers. All right, let's see. This large cube, you can count as done on that big one. The only one we're still waiting on is the large rectangle. This guy right here still has a streak of blue through the middle. It's getting pretty faint. I know it's hard to see in the video. So I will tell you when that one is finished. It'll be not too much longer. There is still a faint little bit of blue in the middle there. Just in case you think I'm not watching anymore, I will let you know when that one is done. We're getting really close. Seems to take the vinegar a really long time to work its way into that last center part. But it is almost there. All right. Oh, still a tiny bit of blue in there. That one's taking forever. All right, it looks like our largest rectangle is finally finished. You may stop your timers on that one. So thanks for hanging out for the cell size, surface area, and volume lab. Feel free to stop into my Google Meets in the mornings or afternoons for these next few days if you have any questions at all. And I hope you have a great rest of your day.